Okay, I thought I'd try to organize things by a sort of user perspective and answer the question, what are the most common questions I've been asked after talks that I've been giving over the last, say, 5 years since we started this work in 2009. And the three most common questions I'm asked are the following. Why should I use an XFEL rather than a synchrotron? Secondly, how do I make the nanocrystals that we need? And thirdly, how does the data analysis differ? Now, why use an XFEL? The important thing to remember is that if you apply to the LCLS, for example, to do an experiment which could be done on a synchrotron, the proposal will be rejected. So it's very important to think of what the new possibilities are with an XFEL. The metric for success of the XFEL is not whether it's a better synchrotron. Your proposals will be rejected if you think that it's just a way to get solved structures more rapidly. The reasons for using XFEL are still emerging. This is early days, of course. But as Paul said, you could think about the avoidance of radiation damage. Now, we detect the early elastic scattering, which starts instantaneously, but of course there's ionization going on, as Paul mentioned, right from the beginning. And I think we're saved to a large extent by Michael Rossman's molecular replacement. Remember, 2/3 of the information comes from the phases, and if you're modeling from the database, that helps a lot. So the purpose of the exercise is to find atom positions, and for that purpose, early ionization of some fraction of your atoms doesn't matter too much. But this is a field of ongoing research, and I'll refer you to some work by Quinney, the Melbourne group, where they have actually seen a charge density wave excited in a crystal of buckyballs by the XFEL, and that produces new Bragg spots. So, as Paul said, we can't simply say there's no damage. I believe, and of course damage is a function of resolution. Personally, I believe that the resolution is perfectly high enough to the extent that we outrun damage to give us useful structural information for proteins without damage. That means that we can study things at room temperature in the native environment by using short pulses instead of freezing to avoid damage. The time resolution obviously is there, whether the biologists want picosecond time resolution. For some problems it's critical, so photosynthesis is an example, but in others the times are much longer, microseconds and seconds, chemical reactions. If the entropy term is important in the free energy rather than the electronic processes, and it's configurational changes that are slow. For trying out new ideas, pump probe work, there's solution scattering, snapshots using single particle work, and so forth, which I'll mention. And in the cases where you can't get crystals large enough to study by conventional crystallography. And then, again, as Paul mentioned, the diffusion into nanocrystals is much more rapid than into a larger crystal. So that allows us to develop things like the mixing jet I'll mention. Okay, I'm going to pass around the bibliography, and that addresses the second question. How do I make nanocrystals? And I've listed there at the end all the papers I could find on how to do it. Now, what's happened is that the protein community has responded to the XFEL by trying to develop methods now, such as diffusion in microchannels and other methods to make nanocrystals suitable for the LCLS and other XFELs. And finally, how does data analysis differ? You've seen the experimental geometry. We spray these nanocrystals. Here we're just talking about nanocrystals across the pulsed X-ray beam and collect about 100 patterns per second. They're in random orientations. So the XFEL is really a terrible source for crystallography. It's a dreadful source because it's not stable. The pulses vary in intensity from shot to shot, up to perhaps 15%. The point of lasing of this thing is jumping around all the time. And there are three main sources of error to keep in mind, which we're all going to be talking about the next 2 days. The unknown orientation of the crystal, we don't have a goniometer. The shot-to-shot variation intensity of the beam, and the size of the crystal. Obviously, if the second crystal is twice as big, you'll get twice as strong Bragg reflections, but that doesn't mean the structure factors are twice as big. So the scaling and normalization is a key issue. There are 4 cases of diffraction we might be concerned with. We're only going to talk in this course about the second one, which I've called big crystals. That means crystals, very roughly speaking, we could argue about this, bigger than about a micron. Now, a lot of the work, the problem we had back in 2009 was people would say to us, how can I make nanocrystals if I cannot see them? I mean, they're invisible. How can you find them and how do you know you've made them if you can't see them under the optical microscope? A lot of our crystals are below the resolution of an optical microscope. And new techniques are being developed now for characterizing nanocrystals, such as the sonic method, and that's referred to in the papers in the bibliography I've given you. So the 4 cases, just to be complete about this, the case of large perfect crystals, like a wafer of silicon. Now, we have a spread of wavelengths in the incident beam, and in this first case of large perfect crystals, not protein crystals, what you find is that the crystal acts as a monochromator. It picks up the wavelength which is at the Bragg condition and sends it off into the detectors. All the other wavelengths go to the beam dump and not diffracted. So the crystal picking out just one is acting as a monochromator. The second case, which we'll be concerned with here, larger as they're called, nanocrystal, bigger than about a micron, is treated by the traditional theory of mosaicity, which imagines that the crystal consists of tiny little blocks slightly tilted. I've drawn them there. What that means then is, roughly speaking, every wavelength goes somewhere. Every wavelength component of the incident beam. Because each of these tilted blocks then can act like a little monochromator at a slightly different Bragg condition. And that's the case that most of us will be concerned with over the next day or two. The third case is nanocrystals, which are really small, just dozens of unit cells across. And for simplicity, let's just imagine we had purely monochromatic radiation. In that case, that single wavelength gets sent into many directions around each spot and causes spot broadening, if you like, particle size broadening, it would be called in powder diffraction. And the final case is what I would call coherent convergent beam. We discussed it at length in our paper in the Phil Trans issue from Royal Society. And that's where you focus down a very small coherent beam, perhaps a tenth of a micron is possible at the LCLS. And then the Bragg spots enlarge into disks because of the large beam divergence, and there are all kinds of possibilities for new experiments there on phasing and so forth. So here are the fringes between the Bragg spots for one of these, a third case there where you have tiny crystals, submicron crystals, which produce interference fringes between the Bragg spots, and I'm calling that shape transforms. So when people talk about shape transforms, they mean these interference fringes running between the Bragg spots that you get from the very smallest crystals, just as you do from a finite optical diffraction grating. And by counting up these fringes, you can tell the size of the crystal. Now, an important point that will be emphasized, I'm sure, today and tomorrow, is that the wavelength spread in the XFEL beam spans wider spots at higher angles. So let's focus now on this case where there's mosaicity, and that is the crystal can be drawn roughly like this, lots of little blocks, each one of which is a monochromator, sending a beam component wavelength of the beam into a slightly different direction. So we need to draw then an Ewald sphere for 2 wavelengths. That's done here. Here's a short wavelength in red and a long one in blue. Here's the origin of reciprocal space. They make different Ewald spheres. Now, what we need to think about is what the reciprocal lattice looks like for a mosaic crystal. Well, each of these blocks makes its own reciprocal lattice, but the blocks are tilted, and so if you imagine a reciprocal lattice vector starting from the origin here and tilted around in all possible angles, or think of the Euler angles, you'll see that it'll trace out a kind of hemispherical cap. So this curved line in green here is a spherical cap at the end of the reciprocal lattice vector. And I've got to do that on every reciprocal lattice vector, so there'll be one up here. If I now impose on that my Ewald sphere, you can see because of the different wavelengths, the gap between the thickness of the Ewald sphere increases with scattering angle, and it'll span perhaps a full range of this spherical cap up here, but much less of it down here. And that's going to produce an error, because we're only measuring part of this angular distribution of this Bragg reflection, but we're measuring all of this one. And that's a key point which the specialists are going to talk about in the next today and tomorrow. So we have to somehow come up with a way of correcting for the errors in what are called partial reflections. And I should say that the pioneering work in analyzing partial reflections, of course, was all done by Michael Rossman's group, and he's here with us today. So if you have questions, he's really the guru on that. So another source So we've said that if we have mosaicity, our reciprocal lattice is smeared out in these spherical caps. What can span that cap? Well, firstly, there's the range of wavelengths in the beam, but secondly, there's the beam divergence, and that's shown here. If we imagine monochromatic radiation covering a range of angles hitting the crystal, then you'll see that that creates a ribbon running across the spherical cap due to mosaicity, and we can define partiality as the ratio of these two areas, the area of the cap divided by, or the area of the ribbon divided by the area of the cap. The ribbon is the spanning effect of the wave vectors across the angular divergence of the beam coming in. Now, all this has been explained, and I mentioned here Michael Rosman's review paper, which this paper in '99 refers to all the early work where the theory was worked out originally. This is taken from Nick Sauter's paper, Hackney, recently came out, and you see beautifully here this effect as the spots, this is experimental data, are moving out in angle, how they get larger, and it's described in detail over here with a detailed construction. So each tilted mosaic block acts as a monochromator for a different component wavelength in the beam, and the energy spread in the beam also thickens the concentric arcs due to mosaicity. You'll have time to come back and look at these later. I can go quickly over some of this stuff because you're going to get copies of all these PowerPoints. And in addition, there is the bibliography I've handed around. Now, do mosaic blocks exist? They were invented as a kind of fiction. It's a highly controversial area where people really, it's a field of ignorance. It's a mathematical model invented way back in the 20s and 30s. Zachariasen has made perhaps a comprehensive treatment of the whole thing, but I would refer you to Ed Snell's review article in 2000, which I've given you the reference to in the bibliography. It gets very particularly tricky for mosaic crystal, for protein, the membrane proteins, I should say. There, I think it just means the defect structure of the crystal. But it's very interesting to me that this Dutch group, Abrahams, has actually, I think, seen mosaic blocks perhaps for the first time. They had the idea of looking with cryo-EM, not at a 2D crystal, or single particles, or tomography, which is a normal way of doing cryo-EM, but at a protein nanocrystal. So you should know that there is now cryo-EM of protein nanocrystals, the ones we study, the XFEL. And the images do show misoriented blocks, and from that you can even determine their size, something which is not known previously. These are the blocks here shown at the top. They don't have sharp boundaries. This is all the result of sampling different regions and different orientations. And I think he says they're about 250 angstroms across. So that's a direct real space. This is a lattice image of many unit cells, not one molecule. Yeah. And there's a group going on at Geneva Farms now, which is trying to solve these protein nanocrystals. With electron diffraction, there are serious problems with multiple scattering, but it'll work fine if they're thin enough. So how do we put all this together? Scaling and partiality, I'll have to let you read this in the cool of the evening tonight. There's too much stuff on this PowerPoint. But people have tried various things, and it's an active field of research. I'll just pick out one point of this. Conceptually, I think it's very important. You should understand that on a single snapshot, you see the angular width of the Bragg reflection sampled at a different point in each Bragg reflection, because the Ewald sphere cuts the same angular profile at different points in each Bragg spot. Yeah? So on one snapshot, you may have all the information to reconstitute the entire angular profile of the Bragg spot, because on every different Bragg spot, it's sampled at a different place. And that's one of the ideas behind the algorithms. Particular, I think Nick Sauter will talk about that, how you can use the union of neighboring spots profiles. And Tom Wise has incorporated similar ideas into his. Another way to go, obviously, is modeling. You can, this is the traditional way it's done, is to describe some kind of peak profile with a few parameters and fit it. And of course, this all breaks down if we have these shape transforms, but I don't think this course today and tomorrow will devote much time to that. However, if you are interested, we did do a treatment of that in this paper back in 2011. Rick Curry and my PhD student worked out a method which we might call dividing out, which will do the scaling and normalization for the case where you see the shape transforms. But iterative schemes are obviously a good way to go, and I'd refer you to a paper that's about to appear by the Cornell group, Phi Delta, where they've used this expectation maximization algorithm for indexing of very small crystals where you just get a few counts in each Bragg spot, and it seems to work magically well. Okay, what can you do? Well, back in 2009, the only thing you can do if you have a really bad system for doing crystallography is to average over all the sources of error. And that's called the Monte Carlo method, which we got from nuclear physics. That's what they do. And it averages, here's the important point, it averages over all sources of stochastic fluctuations. And with a new machine, you've more or less got to do that. I've said there are three important ones: the unknown orientation of the crystal, the unknown size of the crystal, and the shot-to-shot variation of intensity. Now, in the simplest model, we could imagine that they're all Poisson errors and they add in quadrature. And then if you simply add all the Bragg reflections from different crystals together, you'll average over size, you'll hopefully get enough sampling across the angular rocking curve to give you an estimate of the area of the profile of the spot, and you'll average over the shot-to-shot variations. So the idea of the Monte Carlo method, which we started with, was Rick Kieran's PhD, started off with, was just to average over all of those fluctuations by adding all the data together. Does it work? Well, you can tell if it works by plotting the error in this estimated structure factor as a function of the number of shots. And this is Carol Nass's work. It was also a similar plot in our paper in Active Quiz 2011. And you'll see that the error in the structure factors, this is our split in fact, as a function of number of patterns, does fall off like a constant over root n. That's telling us that Monte Carlo is working. Now, of course, everyone wants to do time-resolved diffraction, so the challenge then is to get the error here less than the change in the structure factors due to the pumping illumination in the photosynthesis case. Can that be done? Well, that sets the goal here. Those changes in structure factors can actually be quite large. They can be 15%. So if you have enough data, of course, Monte Carlo will do it for you, but it's very inefficient. And you need 100 times more data to add one significant figure to the results. That's what this law here says. 100 times more data to add one significant figure. So most of the research since 2009 has been trying to improve on Monte Carlo by various forms of modeling. You should know that there are comparisons in the literature between XFEL and synchrotron data. Don't repeat this work until you've looked at this paper. This is the Boutet and Schlichting paper, where in their supplementary information, which people often don't look at, you can see R factors taken between synchrotrons and XFELs. One LCLS and various synchrotrons and lab sources. So these are, this is a good comparison. Now I should say that this was done in the days of, I think, the second generation of detectors. We're now on the third generation, which is much better. So if this were repeated, the results would be better. Again, you'll have copies of this given to you so you can study these in detail later. An important issue is, as Paul mentioned, is access and capacity, how to get time on these machines. One thing which helps a lot is if you can get the data more quickly. And the time to get a complete data set has fallen dramatically. I was at the Gordon conference recently where Helen Jim, a student of Elspeth Garman, reported that she'd collected just 6,000 patterns. This only takes 30 minutes to get a 1.7 angstrom structure. Okay, so with 30 minutes of data collection at the LCLS, she had a structure better than 2 angstroms, 6,000 patterns. And partly that was due to the way she analyzed the data, partly due to the crystal quality, but just the general improvements that have happened in 5 years are quite dramatic, and the biggest of those have been with the detectors. So this compares times to collect data and analyze the data. In our first project back in 2009 with Henry and Tom, it took a year to analyze the data. Now it's done in a few days. Okay, you should know that there is a database, and you'll be using data from this database. Here are 4 sets of serial femtosecond crystallography data that are available on this site. And I've given the in reference to the paper on resolving the indexing ambiguity that will be talked about. I don't have time to talk about background treatment, but that's very important. Other people will do that. And I'll just make a quick comment to end, following up what Paul had mentioned. This LCP jet, I think it's important because it does three things. Here's a membrane protein growing inside this amazing lipid cubic phase, which we collaborated with Martin Caffrey to develop. This is BioXFEL's invention. It's a kind of toothpaste jet where the nanocrystals come out at about the same rate that the X-ray pulses come along. So you don't waste protein. With the liquid jet, which is more general, it could be used for any kind of protein, all the protein between shots runs to waste. Here it doesn't. Secondly, the LCP is a growth medium itself, if you can grow the crystals in LCP. And finally, of course, you don't need so much protein. You need microliters instead of milliliters of stuff. Back in the early days, we needed huge quantities of proteins to use to do structure analysis at the LC layers. So here's this injector. It's a kind of pressure amplifier, not a force amplifier. It's not simple hydraulics, but you need the LCP has a constituency of car grease. Yeah. So you need very high pressures to drive it out the end here. And it's been used to solve a couple of GPCRs, cyclopamine and human serotonin. It works, as Paul said, there's this collaboration now between synchrotrons and XFELs for mutual benefit to both. It works here, and you're seeing the spots in here in this movie appearing where I'm pointing now at the ESRF. And so this is the lipid cubic phase injector running on a synchrotron and giving us nice high-resolution data, but that ring there is the LCP background itself. And here are the conditions which you can check out for yourself on your copies of the PowerPoint. Here's a comparison of different injectors and their behavior and performance. Here's a result from a GPCR that in Chirozov's work with Uwe Weierstahl, where they see differences. Because we're using short pulses instead of freezing to avoid damage, we can look at the structures at room temperature. So it's natural to ask, is the room temperature structure of the protein different from its frozen state? Now, of course, when you freeze samples to put them on the synchrotron, you introduce damage by freezing. But there's less radiation damage as a result of the freezing. On balance, it's believed that you win. And of course, the form of the crystallization depends very sensitively on the chemistry of the crystallization. So side chains can differ just because you did your crystallization, the chemistry of the crystallization differently. But this paper is very interesting, but it compares the room temperature XFEL structure with the frozen synchrotron structure using the LCP jet. Paul mentioned there's been progress with single particles. I'll just bring you up to date on that. This is from Ilma Schlichting's group. It's CRV virus. We did this at the end of last year with the single particle injector. And just to make the point, the resolution here, I reckon, is about 120 angstroms. This is an X-ray diffraction pattern from one virus. Okay? There's only a couple of examples of that in the literature, but most famously the Mimi virus. But we're down to 120 angstroms here. That work in 2009 was 300 angstroms for the Mimi virus. So we've gone from 300 angstroms to 120 in 5 years. Another important result, I think, another growing field will be solution scattering. This time it's snapshot solution scattering, where the molecules don't rotate during the exposure. And I'd refer you to Richard Neutz's paper that's just come out. Here I've shown the difference. This is pump probe, snapshot, solution scattering, right? Don't have to make crystals. And here's shown the difference between the pumped and unpumped reaction center as a function of scattering angle for different times. And what's important is that times are all in the picosecond range. You cannot do this on a synchrotron. It's too fast. What's shown are the difference between the model and the difference measured experimentally. And from this he's able to conclude two important things. That there are fast picosecond motions involved, nuclear motions involved in photosynthesis, which wasn't known before. And that there's a kind of, as they call it, an earthquake in motion, which dissipates the energy from the solar photons without unfolding the protein. So fast solution scattering, I think, is a good name, FSS, fast solution scattering. And I think there's a good, strong future for that. The reason they were able to get such high resolution here, 3 angstroms in solution scattering, is because the ground state was so accurately known. So they had a powerful model from crystallography of the unexcited protein. So this is 3 angstrom resolution, spatial resolution, and picosecond time resolution without growing crystals. That, I think, is remarkable. In my own lab with Jinji Wang, we've built this mixing jet, which may be useful. My job in the SDC is to collaborate with everybody, help everybody use X-ray lasers for protein crystallography. That's what they pay me for. So we made this gadget. It's like a telescope, tubes sliding inside each other. If you think of an enzyme and a substrate, the substrate solution would come in here in B, the enzyme in A, they mix here, then they react for a time, and then we take their X-ray snapshot. And I'm just finished. This is the STC that was funded by the NSF last year. Here are the PIs. It'll probably run for 10 years. And I'm the director of science. Latman is the director of the whole thing. We have a big outreach education program. And the purpose of this is the application and development of BioXFELs to problems in biology. And my job is to facilitate interactions and help people do it. One of the ways we can help you if you want to do this is by making at ASU a test cell, which we'll send to you, probably free. I haven't figured out whether to charge for it or not, which has the sample delivery nozzle stuff in it, and you can test your samples in your own lab before you fly to the LCLS. And we've sent out about 6 of these now. So here's a way to run your samples and see if they work on this our sample delivery system before going to the LCLS. And here's the bibliography I've sent you. I forgot, Michael Rossman's paper is not here. It's given in an earlier PowerPoint. That's it. Any questions? Yeah, Tom.
A comment regarding the number of patterns used for different solutions and how it's changed. People doing these experiments rarely test how many they really need, so how few patterns they can get away with. The numbers in the papers normally reflect the amount of data that has been obtained. So so there's 6,000 for that experiment they really investigated. So what
is that? Is that the number of indexable patterns, or is it the total number of patterns?
That's the number they used. I think they said with fewer they couldn't get the structure.
So this is after index. This is the number that they could index.
The number that have gone into the number merged.
Right. Yeah. So patterns with less than about 20 spots don't index.
Yeah. Actually not as far as I understand it, with that experiment, 6,000 is just a subset of all the data they collected. It was a very special crystal, but I mean, this is relatively important from
Yeah, but we're agreed that this is the number they could index, not the number they collected.
No, that was a subset.
But this is the number they collected.
No, this is a subset of all those.
Yeah, that's what I said.
We indexed about 75,000 patterns. And
were you involved in this project?
Yes, I was.
Okay, good.
We indexed about 75,000.
Yeah.
Alan simply took the first 5,000 or 6,000.
And that was enough for a complete set. Great.
The first 5,000 and not the best 5,000.
Yes.
Okay, that's important.
Yeah, it's different.
The point I was really making is this number here, 170, left one cell, up that one. This number is not the number that was, quotes, needed. This was just the number that we had because we got very lucky with the sample delivery and the sample worked fantastically well. So we had a lot of data, and of course we used it to get the best structure we possibly can. Right. But this would have worked with, I think we tried briefly with 11,000 and that worked absolutely fine as well.
Don't you think, Tom, the fact that people are publishing structures below 2 angstroms means that crystal quality is still the limiting factor, not the radiation damage of the XFEL or any intrinsic resolution limits of the method?
I'm not so sure.
I might just take this opportunity to say, can we continue this during the panel discussion?
Yes.
Okay, Michael Russell.
I want to comment on the necessity. In days gone by, we used to talk about perfect and imperfect crystals.
Yeah, ideally imperfect.
Ideally perfect. So an ideally perfect crystal is like diamond, which is a single lattice and not just one single block. Under those conditions, the individual deflected X-rays interfere constructively and destructively. If the mosaic blocks, and then the relationship between the intensity and the measured the structure factor is a linear relationship. But if they are in different blocks, then the individual mosaic blocks each to their own deflection, and you just add up the intensity. There's a squared as opposed to an F relation.
I quite agree, and I left it to Holton to show Darwin's formula, which he has. I mentioned Terry Sabine's work. The complete theory of extinction involved multiple coherent scattering within a block and incoherent multiple scattering between different blocks. And the whole thing gets very complicated, as you know.
Exactly.